Hi students, myself Chandrasekhar, Zoology faculty from Sri Chaitanya. In Sri Chaitanya's NEET Sankalp 2.0, we are moving forward with the next chapter in zoology, that is locomotion and movement, which is a very important chapter in physiology. So without making any late, let's get into the topic. See, the first point here is movement is one of the significant features of living beings. The point is both animals and plants exhibit a wide range of movements. For example, if you take the unicellular organisms like amoeba, amoeba can move with the help of pseudopodium, which is caused by the streaming of protoplasm. Paramecium, it shows ciliary movement. Euglena shows flagellar movement. Even the hydra shows the movement of tentacles. The important point regarding it is, we can move our hands, we can move our tongue, we can move our eyelids. But when we move our lower limbs, it causes the displacement of the organism. So the displacement of the organism is said to be locomotion. The important point I want to discuss here, locomotions are movements, but all movements are not locomotion. See, some movements may not result in displacement, we can't call it as locomotion. And one more important point here is, locomotory structures need not be different from those affecting other types of movements. See, if you talk about paramecium, in paramecium, the cilia helps in the movement of organism, the cilia helps in the movement of cytopharynx inside the body, which causes the digestion of food. So that means it is helping in locomotion as well as food collection. When we talk about pseudopodium, pseudopodium helps in locomotion and food collection. Tentacles that are present in the hydra, they are helping in both locomotion and food collection. So that is the reason why we can say locomotory structures need not be different from those affecting other types of movements. The one more important point regarding this is, methods of locomotion performed by animals vary with their habitats and the demand of the situation. See, the aquatic organisms movement is swimming, but whereas when it comes to the terrestrial organisms movement, it is quite different from that. So that is the reason why the type of habitat where the organisms are living that can influence the movement of the organism. And what is the main purpose of this locomotion? The purpose of locomotion is generally for in search of food, shelter, mate and suitable breeding grounds and also to escape from the predator. So these are the important components that are required, locomotion is required to perform these functions. And regarding amoeba, I just want to tell you, see the amoeba, so these are the pseudopodia in amoeba, which is caused by the streaming of protoplasm. If this is paramecium, these are the cilia helping in locomotion. If this one is euglena, this one is a flagellum involved in locomotion. In hydra, these are the tentacles helping in locomotion as well as food collection. And these first three are unicellular organisms, they belongs to the kingdom Protista. But hydra is not a unicellular, it belongs to the phylum Cnidaria and the kingdom is Animalia. So this is the introductory part regarding locomotion and movement chapter. Let's talk about types of movements. Cells of the human body exhibit three main types of the movements. The first movement is called amoeboid movement. If it is a macrophage, see, if it is a macrophage, this is the cytoplasm. The streaming of the cytoplasm can cause the temporary outgrowth is said to be pseudopodium. So the movement performed with the help of pseudopodium is called amoeboid movement. In our body, amoeboid movement is exhibited by the macrophages and leukocytes. So human body cells that are showing amoeboid movement come under macrophages and leukocytes. And not only the streaming of cytoplasm, even the microfilaments are also involved in the formation of pseudopodium in human beings. The second type of movement is ciliary movement. Wherever the ciliated epithelium is there you can see the ciliary movement. For example, fallopian tubes, they are having the cilia, the movement of cilia there causing the movement of ovum. And when we talk about trachea, the trachea are having cilia. Those cilia, upward movement of cilia can able to trap and move the dust particles towards nose. So, elimination of dust particles. And even our brain and spinal cord, we have ciliated epithelium that causes the currents in cerebrospinal fluid. So, ciliary movement is there in these parts like fallopian tubes, trachea, bronchi, central canal of spinal cord, ventricles of brain. And the third type is called flagellar movement. If you see the structure of human sperm, the human sperm consists of head, neck, middle piece, and the final part is called tail. The tail consists of axial filament. The structure of axial filament consists of 9 peripheral doublets and 2 central ciglets, and that is equal to the structure of flagellum, and the type of movement performed by the human spermatozoon is said to be flagellar movement. And when it comes to the main types of movement in human beings is muscular movement. See, with the help of muscles, we can able to move our limbs, jaws, tongue, etc. So, the main type of movement in human body is muscular movement. Muscular movement is caused by the contractions and relaxations of muscles. So, let us talk about muscle. 40 to 50% of our body weight is constituted by the muscles. This point I have already discussed in the structural organization. And muscles are having 4 basic properties like excitability, that is nothing but giving response, contractility, elasticity and extensibility. These are the properties of muscles. Muscles are derived from mesoderm, but there are some muscles that are derived from ectoderm. The examples like ciliary and iris muscles. And there are different classifications of muscles. Depending on the location, we can classify the muscles like skeletal muscles, visceral muscles, cardiac muscles. Depending on the mode of control, you can classify the muscles into voluntary and involuntary muscles. Depending on the appearance, you can classify the muscles into striated and unstriated muscles. Here, based on the location, let us discuss the types of muscles. One is skeletal, second one is smooth or visceral, and third one is cardiac muscle. Let us see the characters of or differences between skeletal muscle, smooth muscle and cardiac muscle. See this picture. The first picture is related to the skeletal, second is smooth and third one is cardiac. This one is skeletal, this one is cardiac. Okay, let us talk about the characteristics. Regarding the structure of a muscle, the skeletal muscle occur in the form of bundles. The entire muscle is covered by a connective tissue sheath known as fascia. I will give you the structure of skeletal muscle separately. And each skeletal muscle fiber is long and cylindrical. Each skeletal muscle fiber showing the dark and light bands in alternate way, showing the striations, hence it is called striated muscle. And regarding the contraction control, central nervous system can regulate the contractions of skeletal muscles through somatic nerves. The skeletal muscles are said to be voluntary muscles. The skeletal muscle fiber is having many nuclei in each cell, hence it is said to be syncytial nuclei condition, so that we can call it as structural syncytium. And the anatomical unit of a muscle is muscle fiber. Anatomical unit is muscle fiber. And about the examples of skeletal muscles includes are bicep muscles, tricep muscles, muscles of tongue, muscles of abdominal wall, diaphragm, they are all coming under skeletal muscles. And skeletal muscles can contract quick and fatigue quick. So, this is about skeletal muscle points. When it comes to the smooth muscles, smooth muscle is also covered by the connective tissue sheath, but it is not fascia. They are bundled together by connective tissue sheath, and between smooth muscles there are cell junctions. They held together by cell junctions. Each smooth muscle cell is fusiform, sharp at both the ends. Also you can call it as spindle shaped, uninucleated, and they are not having any striations, hence they are unstriated in nature. And I am telling you guys, unstriated means it is not absence of myosin and actin. All muscles are having actin and myosin. Unstriated means the alternate appearance of dark and light bands is absent in smooth muscles. Next, regarding its appearance and the parts where the smooth muscles are located. Smooth muscles, they are called smooth due to the absence of striations, and they are having another name called visceral muscles because these muscles are located in the walls of visceral organs, like in the wall of digestive tract, wall of respiratory tract, wall of circulatory tract, that means wall of blood vessels, wall of excretory tract, urinary bladder, wall of reproductive tract, uterus in females. So, they are all having smooth muscles. Even the ciliary muscles and iris muscles of eye are also comes under smooth category. And when they are located in the visceral organs, we can call them as visceral muscles. The third category is cardiac muscles. Cardiac muscles occur in the form of branches. They are branched. See, in cardiac muscles, there is a specific cell fusion point is present known as intercalated disc. Assume this is one more intercalated disc. Between two intercalated discs, whatever the portion is present in a tube, you can call it as muscle cell, myocyte. And they are uninucleated, they are having striations striated, and they are involuntary in nature. See, smooth muscles and cardiac muscles are involuntary because they are not under the direct regulation of nervous system, they will be regulated by the autonomic nerves. Both sympathetic and parasympathetic nerves controls the contractions and relaxations of smooth muscles and cardiac, hence they are said to be involuntary in nature. What about the cell fusion junctions? So, these intercalated discs are having gap junctions. The gap junctions will allow the rapid movement of small molecules and even sometimes large molecules and ions. This rapid movement of ions is involved in rapid information exchange, hence they are called communication junctions. Because of gap junctions, whatever the action potential generated in one muscle cell can easily spread to the other muscle cells, making the whole muscle as a unit to contract. That is the reason why the cardiac muscle is said to be functional syncytium. It is said to be functional syncytium. Examples where the cardiac muscle is located, it is present in the wall of the heart, it is also present in the specialized musculature forming the nodal tissue, like sinoatrial node is having cardiac muscle, atrioventricular node, even our Purkinje fibers present in the heart are made up of modified cardiac musculature. So, these are the differences between three types of muscles. Now, let us get into the skeletal muscle story. So, regarding the skeletal muscle, see this, they have got the name skeletal because they are attached to the bone. They are attached to the bone. Skeletal muscles are attached to the bone with the help of a dense regular connective tissue, known as tendons. Entire skeletal muscle is covered by a connective tissue sheath known as fascia, and inside the fascia, that means inside the skeletal muscle, there are many bundles present called fascicles. Muscle bundle is called fascicle. Each fascicle is covered by a layer called perimysium. This is also connective tissue sheath. But one fascicle consists of many muscle fibers. Fibers are nothing but muscle cells. Muscle cell is also known as a sarcocyte. And the connective tissue sheath covering the muscle fiber is called endomysium. The plasma membrane of the muscle cell is called sarcolemma. And one muscle fiber consists of many other components, longitudinal components inside, they are called myofibrils. So, when you want to arrange the larger to smaller, a muscle consists of fascicles, fascicle consists of fibers, fiber consists of fibrils, and inside the fibrils there are many filaments present. This is the largest to smaller organization in a skeletal muscle. Fascia is covering the all fascicles, perimysium is covering one fascicle means one bundle. See, this is the NCERT picture. So, this whole thing is fascicle. Inside these pink color structures are muscle fibers. Muscle fibers are covered by a plasma membrane called sarcolemma. Where there are blood vessels are there, we can say muscles are vascular in nature. So, let us see structure of a muscle fiber. In one muscle fiber, Assume it is one muscle fiber, long and cylindrical. The membrane is called sarcolemma. Cytoplasm present inside the muscle cell is called sarcoplasm, and peripherally located syncytial nuclei. It has cell organelles. Of course, there is an important protein inside called myoglobin. It is red in color. The function is oxygen storage. Then several myofibrils present. There is endoplasmic reticulum inside, mitochondria. Of course, for the energy production there is an ATP and other minerals like sodium, potassium, calcium present inside. So, this is the structure of a fiber. Let us see the structure of a fibril, very important. So, what is there inside a fibril? Inside a fibril, see this. Inside a muscle fibril, there are many filaments present. This dark color filament is called thick filament. These light color filaments are called thin filaments. Thick and thin filaments. So, this length of one thick filament area, including all the parts inside, is said to be A band. A band is also called a dark band. It has got the name A because it is anisotropic in nature. That means it is more darker towards the end of the A band, it is less darker at the center. At the center of A band, just above the thick filament, this is the area, it is not overlapped by the thin filaments. This non-overlapped area above the thick filament is called H zone, which is also called Henson's disk. It is made up of only thick filaments. And at the center of a H zone, there is a membrane separating it into two halves, that is called M line. M line is made up of collagen fibers. This is regarding A band. A band is having thick and thin filaments. Just above the thick filament at the center of the A band, non-overlapped area is called H zone. At the center there is a membrane dividing it called M line. M line is made up of collagen fibers. So, it has thick filaments and thin filaments forming A band. And see the peripherals. On either sides of A band, the next one is called I band. I band is made up of only thin filaments. See these light color structures are thin filaments. This is a thin filament, thin filament, thin filament. And each I band is bisected into two halves by a Z membrane. Z membrane is made up of elastic fibers. It is elastic in nature. See, this is one I band Z line. This is another I band Z line. The area present between two successive Z lines is known as a sarcomere. This sarcomere consists of one A band and two halves of I bands. And sarcomere is said to be the contractile unit of the muscle. It is the functional unit of the muscle. Anatomical unit of the muscle is muscle fiber. Sarcomere is the functional unit. So, this is regarding the structure of myofibril. And let us see the structure of myofilaments. I will open this thick filament, I will show you what is there inside it. I will open this thin filament, I will show you what is there inside the thin filament. So, regarding the structure of filaments, see, this is a thin filament. A thin filament is made up of three components. F actins, but there are two F actins, these green color structures. This is F actin and this one is also F actin. Two F actins. On the F actins, there are sites to bind with the myosin. If I put dots over this, these are the sites of actin to bind with the myosin. They are called active sites, also known as myosin binding sites. Next, covering those, there are two filamentous proteins present in the thin filament known as tropomyosin. So, on these tropomyosins at regular intervals, another component called troponin is present. See, each troponin is having three circles. So, this is troponin first circle, second circle, third circle. Tn for troponin. So, TNC, TNT, TNI like that. So, calcium binding site of troponin, tropomyosin binding site of troponin, the subunit which is acting as inhibitor. There are three subunits in one troponin and it is present at regular intervals on the actin molecules. When the muscle is at rest, when the muscle is at rest, a subunit of troponin, it masks the myosin binding sites on actin. It covers it. It will not expose those sites when the muscle is at rest. But at the time of contraction, the unmasking of these sites and exposure of these sites can be seen. So this is about thin filament, 2 effactins, 2 tropomyosins and many troponin molecules. Let's talk about thick filament structure. So regarding the structure of thick filament, see this. A thick filament consists of 200 to 300 myosin monomers. This is the structure of a myosin monomer. Such kind of myosin monomers, how many present in one thick filament? 200 to 300. Hence, thick filament is a polymerized molecule. Of course, thin filament is also polymerized because F-actin consists of many circles, green color. Each circle is called G globule, G molecule. So it is also a polymerized molecule, and thick filament is also polymerized molecule. And each a myosin monomer, it has 3 parts like head, neck, tail. The neck and tail together called, neck and tail. Neck is called short arm. Head, neck together known as a cross arm. It is called cross arm because it shows orientation at regular intervals. Hence it is said to be cross arm. And in the head region there are sites. The site of myosin which is there to bind with the actin is called actin binding site. See, actin is having myosin binding site, active site. Myosin is having actin binding site. Next, the myosin is also having site to bind with the ATP, which is called ATP binding site. So 200 to 300 myosin monomers are forming one thick filament, myosin filament, which is also known as, and having parts like head, short arm, that is neck and tail. And when we break the myosin monomer, actually it is a protein by using the proteolytic enzyme at the neck region, at this particular region, we can divide the myosin monomer into cross arm and tail. As cross arm molecular weight is more, it is said to be heavy meromyosin. This cross arm is heavy meromyosin and the tail is called light meromyosin. So meromyosin, with the help of enzymes like trypsin, it can be broken down into HMM and LMM. So this is the structure of contractile proteins. And in this, actin and myosin are called contractile proteins because they generate force during contraction. Troponin and tropomyosin are called regulatory proteins because they are involved in the masking and unmasking required for the contraction. Next, the next topic is mechanism of muscle contraction. Mechanism of muscle contraction is best explained by a theory called sliding filament theory. Sliding filament theory. In sliding filament theory, the thin filaments slides upon thick filaments. Actin filaments slides upon myosin filaments. And in the sliding filament theory, initially the ATP is broken down and provides energy for the movement of the muscle. Hence, chemical energy is converted into mechanical energy in sliding filament theory. And regarding the steps of sliding filament theory, the first step is excitation. And I am talking about skeletal muscle contraction. That means I have to talk about CNS sending signals to the somatic motor neurons. So the first step is excitation. See this, if it is CNS, with the help of somatic motor neuron, it gives signals to the muscle fiber. Assume it is muscle fiber. This one is sarcolemma. This is called neuromuscular junction. So this is motor neuron, this is muscle. Motor neuron and muscle together called motor unit. But the junction between motor neuron and muscle is called neuromuscular junction. And these are axon terminals having synaptic vesicles and neurotransmitter are called acetylcholine. The first step is excitation in the contraction, muscle contraction mechanism. When CNS gives the signals to the somatic motor neuron, that will reach the axon terminal. There it causes the release of neurotransmitters into this junction. The most common neurotransmitter is acetylcholine. It will go and bind to the receptors present on the sarcolemma. When a neurotransmitter binds to the receptors on the sarcolemma, immediately it causes depolarization of the membrane of muscle called sarcolemma due to the influx of sodium. That depolarization will cause or responsible for the release of calcium from the cisternae of sarcoplasmic reticulum into the sarcoplasm. When calcium is released, you already know that there are thick and thin filaments in the sarcoplasm. The thin filament consists of troponin. Troponin is having a subunit called TNC. Now the calcium will go and bind to the TNC subunit. Calcium will go and bind to the TNC subunit. Once the calcium binds to the TNC subunit of troponin, listen, this is the Z line, and between two Z lines this is the sarcomere, and this one is the thin filament, assume, and this is the thin filament. Tropomyosin and troponin, tropomyosin and troponin. Now the calcium bind to this. The binding of calcium will make some conformational changes in the troponin and as well as tropomyosin. That causes unmasking of myosin binding sites on actin, and this one is myosin. Unmasking. At this moment, I already told you myosin head consists of ATP binding sites. ATP will bind to the myosin head. The ATP will be hydrolyzed. Hydrolysis of ATP forms ADP and inorganic phosphate. That gives energy to the myosin head. Now the myosin head will move to the active site of actin and holds it, and it pulls the actin filaments towards the center of A band. That is called contraction. See, first step is excitation, in which neurotransmitter released, then sarcolemma depolarized, calcium released will go and bind to the TNC. After that, unmasking, because calcium causes the changes in the troponin and tropomyosin, and unmasking the myosin binding sites on actin, followed by ATP binding, ATP hydrolysis, giving energy, then myosin holding the active sites and pulling towards the center of the A band. So when it is pulled towards the center of A band, that is called contraction. So what happens during the contraction of muscle? What are the structural changes that can occur in the sarcomere? Let me explain this. See this? This is one sarcomere in relaxed state. When myosin, this is the thick filament, when actin filaments, thin lines are pulled towards the A band, what happened to the H zone from the first picture to the second picture? The H zone compared to the first picture, it is decreased in the second picture. So there is decrease in H zone during contraction. This is the sarcomere of first picture, this is the sarcomere of second picture, there is decrease in the sarcomere. This is the A band of the first picture, this is the A band of the second picture, sorry, I band of the first picture, and this is the I band of second picture, I band reduced. So during muscle contraction, H zone decreases, sarcomere decreases, I band decreases, but there is no decrease in the length of the thick filament, here and here same. There is no decrease in the length of the thin filaments, here and here same. Thick and thin filaments remain same, and even the length of one thick filament is equal to the A band. A band also will not change, it remains same. So this is very, very important. Structures that are undergoing changes, structures that remain same. H zone decreases, I band decreases, sarcomere decreases. Thick filament, thin filament, A band remains same during contraction. And in a maximally contracted muscle, when the myosin heads, when they pull the actin towards the center of the H zone with much force, see in the third picture, there is no H zone here. In a maximally contracted muscle, H zone will disappear. Not only H zone, on either sides of the Z line, there is no I band also will disappear in a maximally contracted muscle. So this is regarding contraction. Contraction is followed by the relaxation, guys. And what is relaxation? Whatever the events I told you so far, they need to get reverse. That means CNS stops sending signals to the neuromuscular junction and to the muscle. That is one event during relaxation. Listen carefully. So, during relaxation, see, CNS stops signaling. Calcium which is moved from cisternae to the sarcoplasm, it will go back to the cisternae. Unmasking become masking. There is breakage of cross bridge, and Z lines move to their original positions. Sarcomere gets normal. So, these are the events of relaxation of muscle. Contraction followed by relaxation. In NCERT, they have mentioned about events of one contraction cycle in a picture. Let me explain this. These events are very, very important, guys. See, in this picture, see, this is thin filament, this is the cross arm, myosin head, thick filament story. I will give you the sequence of events in one contraction cycle. Binding of ATP is the first event. This binding of ATP to the myosin head, it causes the breaking of a cross bridge of the previous cycle. So, event 1 is binding of ATP, event 2 is, see, there is a gap between thin and thick filament, cross bridge breaks. Second event is breaking of cross bridge. The third event is the ATP undergo hydrolysis into ADP and inorganic phosphate, that is event 3. Once ATP is hydrolyzed, myosin head gets energy. The head, see, it is moving up and attached to the actin filaments at the active site, forming a cross bridge. Formation of cross bridge is the next event. Once cross bridge is formed, see the straight here, now it is causing the rotation. Rotation means pulling it, causing a contraction. That is the next event, which is also called sliding, followed by whatever the ADP hydrolyzed. Now with this ADP will get detached, providing space for the attachment of new ATP and new cycle. So, that is the last event. Remember these 6 events in sequence, guys. The first event is binding of ATP, second event is breaking of cross bridge, third is hydrolysis of ATP, fourth is formation of cross bridge, fifth is sliding or rotation, followed by the detachment of ADP. So, these are the events related to one contraction cycle that are explained in this picture. Next, so with this muscle contraction mechanism completed. The next topic is types of muscle fibers. There are 2 types of muscle fibers, like red muscle fibers and white muscle fibers. Red muscle fiber and white muscle fiber. I want to give you the differences between them in a quick note. Red and white. So, regarding red, first I will write the points. They consist of more amount of myoglobin, consists of more oxygen, mostly perform aerobic respiration, fatigue slow because of more oxygen. See, more myoglobin, more oxygen, mostly aerobic and fatigue slow, and they consist of more amount of mitochondria because it performs aerobic respiration. When it comes to the white muscle fibers, compared to the red, less myoglobin is present here, less amount of oxygen because myoglobin stores oxygen, as it is less myoglobin, less storage of oxygen, and it is mostly involved in anaerobic respiration, cellular respiration, fatigue quick. I will tell you what is fatigue, do not worry, and they consist of less mitochondria. But one more point I want to tell you here, regarding the sarcoplasmic reticulum, red muscle fibers are having less sarcoplasmic reticulum, white muscle fibers are having more sarcoplasmic reticulum. And the contractions of red muscle fibers are slow and low intense, and contractions of white muscle fibers are quick and high intense. And red muscle fiber is said to be slow oxidative muscle fiber, and white muscle fiber is said to be fast glycolytic muscle fiber. and more storage of glycogen can be seen in the white muscle fiber compared to the red muscle fiber. And what is muscle fatigue? Let me tell you now. Anaerobic breakdown of glycogen leading to the accumulation of a substance known as lactic acid. So, this lactic acid, it causes the hardening of the muscle. That condition is said to be muscle fatigue. Anaerobic breakdown of glycogen leading to the accumulation of lactic acid causing a condition called muscle fatigue. And when do this lactic acid is produced? Anaerobic. Who is mostly depending on anaerobic? White. So that muscle fibers get fatigued fast when compared, white muscle fibers fatigue fast compared to the red muscle fibers. So this is about the differences between red muscle fiber and white muscle fiber. And one point I want to tell you, like the fatigue muscle will come to the normal state by cyclical events known as a Cori cycle. I am not explaining this, just giving you one extra point. Cori cycle which is involved in normalizing the fatigue muscle to a normal state. One more point is muscle twitch. Muscle twitch is nothing but a brief contraction of all the muscle fibers in a motor unit with a single signal. For example, if you take one motor unit consists of many muscle fibers. See this? This is one motor unit, axon terminals and many. Single signal will cause the contraction of all these muscle fibers at a time. That brief contraction of all the muscle fibers in a motor unit is said to be muscle twitch. So with this, the muscle topic is completed. The next topic in our chapter is the skeletal system. The skeletal system consists of framework of bones and a few cartilages. We already know that the functions of bones includes, they form the structural framework of an organism. They support and protect the soft tissues of our body, involved in locomotion, homeostatic reservoirs of minerals, and in long bones, the red bone marrow is involved in the production of red blood cells. And when it comes to the cartilages, the difference between bone and cartilage, both are having collagen fibers, but one is having calcium salts bone, one is having chondroitin salts cartilage. Cartilage is pliable, bone is non-pliable, which is rigid in nature. Cartilage, there is a point like it is pliable and resists compression. Even bone can resist the compression. And cartilages are located at the tip of the nose, nasal septum, ear pinna. Next, intervertebral disc between vertebrae is made up of fibrous cartilage. Pubic symphysis, cartilage between ribs and sternum, costal cartilage. Epiglottis is made up of elastic cartilage. Our larynx is made up of cartilages. They are all the examples for cartilages, and they are having chondroitin salts. Human skeleton, generally the adults consist of 206 bones. We can divide it into two categories: axial skeleton, appendicular skeleton. So the skeletal parts that are there in the axis, longitudinal axis on and around is called axial skeleton. Like skull comes under axial skeleton, vertebral column backside comes under axial skeleton, sternum axial skeleton, and ribs also comes under axial skeleton. And when it comes to the appendicular skeleton, they are limbs and girdles. Pectoral girdle supporting upper limbs, pelvic girdle supporting lower limbs comes under appendicular skeleton. Out of 206 bones, 80 bones comes under axial skeleton, 126 bones comes under appendicular skeleton. Let us talk about the skeletal parts one by one, starting with the axial skeleton. The first part is the skull. The skull consists of two sets of bones, like a cranium and facial bones. Cranial bones are 8, facial bones are 14, 8 plus 14, 22. The number of bones in skull having the two sets of bones are 22. When it comes to the cranial bones, listen carefully. Cranium is a bony box which can protect the brain, and cranium is made up of 8 bones. There is a bone which is forming the forehead known as a frontal bone. The upper surface of the cranium is formed by the two parietal bones. This is one parietal, other side one, but frontal is only one. The posterior region of this cranium is formed by the occipital bone. This occipital bone, backside it has two condyles called occipital condyles. Because of the two condyles, our skull is said to be dicondylic in nature. And for this occipital bone, there is an aperture called foramen magnum. Through that aperture, the medulla oblongate of the brain will continue as a spinal cord. Next, laterally we have two temporal bones. This is one temporal, other side one more. Next, there is a bone called ethmoid. Behind that there is a bone called sphenoid. So this is ethmoid bone, this is sphenoid. These are the bones of cranium. So list of bones in cranium includes one frontal, first unpaid bones I am telling you, one frontal, one occipital, one ethmoid, one sphenoid, 4, and two parietals, two temporals, 4, 4 plus 4, 8, unpaid bones 4, paid bones 2. And regarding the functions, frontal is forming the forehead region, upper region is formed by the parietal, lateral regions is formed by temporal. Through the temporal only the auditory canal can pass. And regarding the ethmoid and sphenoid, especially this sphenoid bone is said to be the keystone bone because it is in contact with the all other bones of the cranium. So this is enough. And between the cranial bones in the skull, there are joints called sutures present. Sutures comes under fibrous joints. They are made up of dense fibrous tissue and they are the immobile joints. The suture between frontal and parietal is said to be coronal suture. The suture between parietal and temporal is called squamosal suture, and the suture between parietal and occipital is called lambdoid suture. So this is about cranium. And the next set of bones are facial bones. See, front side bones. Regarding the facial bones, listen guys, there are 14 bones in the facial region. Just I will give you the names. It is not there in NCERT textbook, but some bones are mentioned in the diagram, so that I want to give you the names of facial bones. See, in the orbits of our eye, we have a pair of lacrimals, one pair of lacrimal bones. A rough sketch I will give you. So regarding the face region, these are the orbits, a pair of lacrimal bones, and in between we have a pair of nasals. Then we have a bone forming the floor of the nasal cavity called vomer. Two bones forming the lateral walls of the nasal cavity called inferior conchae. L for lacrimals, N for nasals, V for vomer, and inferior conchae, I for. Then a pair of cheek bones present known as zygomatics. Then this is the lower jaw bone, this one is the upper jaw bone, and behind that we have a pair of palatines, P for palatines. Upper jaw bones are called maxilla, lower jaw bone is called mandible. Listen carefully, one pair of lacrimals 2, one pair of nasals plus 2, 4, one pair of inferior conchae 6, one pair of palatine 8, one pair of maxillae 10, one pair of zygomatics 12. These are all paid. Unpaid bones is one vomer and one mandible. So mandible is the largest bone of the facial region. Mandible is the only mobile bone of the face, and it is the strongest bone of the face. Among the facial bones, the smallest is lacrimal. So there are 14 bones, out of which 2 bones are unpaid, vomer and mandible. Remaining bones are paid: lacrimals, nasals, zygomatics, inferior conchae, palatine bones, maxillae. Maxillae means upper jaw bones. So these are the facial bones. Next, let us talk about so these two bones constitute the skull. The 8 plus 14, 22 bones are there in the skull. Next is related to hyoid bone. Hyoid bone is also U-shaped. It is present just above the larynx, voice box, at the floor of the buccal cavity, and it keeps the larynx open, and even the tongue muscles are attached to the hyoid bone. Hyoid is U-shaped just above the larynx, present at the floor of the buccal cavity, and tongue muscles are attached to the hyoid bone. This is the hyoid. The next is ear ossicles. Each ear consists of 3 ear ossicles starting from within the middle ear between tympanum and the oval window. Malleus, incus, stapes. These are the 3 ear ossicles. 3 in one ear, 3 in another ear. So 6 ear ossicles are there. So with this we have completed cranial bones, facial bones, hyoid and ear ossicles. But cranial and facial are related to the skull bones. The total number of skull bones here are 22. Next is, after skull, the next one is vertebral column. See, vertebral column, it is curved. There are 4 curves like cervical curve, thoracic curve, lumbar curve and sacral curve. Vertebral column is made up of series of bones called vertebrae. There are 7 vertebrae in the neck region, cervical vertebrae C1 to C7. There are 12 vertebrae in the thoracic region, 5 in the lumbar region, 1 sacrum, 1 coccyx. These are in adults, but in embryonic stage, sacrum is not 1, 5 sacral vertebrae present. They fuse it to form 1 sacrum here, which is triangular in shape. This is the lateral view, but with the front view we can say triangular bone. And the last one is coccyx. Coccyx is single in adults, but coccyx is also formed by the fusion of 4 coccygeal vertebrae. 26 vertebrae present in adult, whereas 33 in the embryonic stage. Okay. Regarding the cervical vertebrae, the first cervical vertebrae is named as atlas. The second cervical vertebrae is called axis. Let me tell you one point here. If it is the skull, these are 2 occipital condyles, back side of the skull. Assume this one is atlas, this is axis, atlas, axis. Between occipital condyles and atlas there is a condyloid joint. See, nodding of head, we can use condyloid joint. Between atlas and axis there is a pivotal joint, rotation of head. Both are synovial joints only. Next, if you see the basic structure of one vertebra, I am going from front side to the back side of vertebra, but I am showing you the lateral view. So there is a part called centrum in each vertebra. Assume this is centrum, followed by neural canal, followed by neural spine. Again this is centrum, neural canal, neural spine. Centrum, neural canal, neural spine. And who is passing through the neural canal? Spinal cord. Spinal cord is passing through the neural canal. And what is there between this centrum? Between centrum to centrum, there is a cartilage present known as intervertebral disc. This is intervertebral disc. Intervertebral disc is forming the cartilaginous joint which allows a limited movement. It is present between centrum to centrum between the successive vertebrae. So this is regarding the basic structure of vertebra, and first vertebra atlas, second vertebra is axis. And let us see the functions of these vertebrae. Cervical vertebrae, they are involved in the movement of the head and supports the head. Thoracic vertebrae, see this. Our thoracic vertebrae present at the back side, they provide surface for the attachment of ribs. All 12 pairs of ribs attached to 12 thoracic vertebrae. That means each thoracic vertebra is articulated by a pair of ribs. So thoracic vertebra 12, total ribs are 12 pairs. Next, lumbar vertebra supports the back muscles, sacrum is supporting the pelvic girdle, and coccyx has no particular function. So these are the functions related to the vertebral column. Intervertebral disc, I told you already, cartilaginous joint. Next, numbers are also important: 7 cervical, 12 thoracic, 5 lumbar, 1 sacrum, 1 coccyx. The sacrum is triangular in shape, formed by the fusion of 5 sacral vertebrae. Let us talk about another bone called sternum. Sternum is a single bone which is also called breastbone. Sternum is present on the mid-ventral line in the thoracic cavity. It is providing surface for the attachment of true ribs. See, the first 7 pairs of ribs are attached to the sternum. They are called vertebrosternal ribs, nothing but true ribs. Sternum also provide a surface for the attachment of front abdominal muscles. This is a dagger like bone, and it has 3 basic parts, like upper part of the sternum is called manubrium, middle part is called body, and inner part is called xiphoid. And there is a collar bone called clavicle. Clavicle towards the median side of the body, it is attached to the manubrium of the sternum. So, clavicle is attached to sternum, true ribs are attached to the sternum, and front abdominal muscles are also attached to the sternum. And the third one is ribs. There are 12 pairs of ribs. Ribs are flat bones, long bones. Ribs are said to be double-headed, bicephalic ribs, because it has 2 articular surfaces towards the dorsal side of the body. It is not on the ventral, towards the dorsal side, while the rib is attached to the vertebra. It has 2 head-like structures. With one head, it is attached to the centrum of the vertebra. With another head, it is attached to the transverse process of the vertebra with the help of capitulum and tubercle. Next, we can classify the ribs into true ribs and false ribs. First 7 pairs of ribs are called true ribs. Back side they attach to vertebra, front side they attach to sternum. They are called vertebrosternal ribs. Next 5 pairs are false ribs, but we can divide the false ribs into 2 categories. After 7, 8th, 9th, 10th are called vertebrochondral ribs. Back side they attach to vertebral column, front side they attach to the cartilage. See, this blue color structure is a cartilage, hyaline cartilage, which is present between ribs and sternum. So, 8th one, 9th one, 10th one, they are attached with each other at the cartilage site, hence they are called vertebrochondral. 11th and 12th ribs, they are attached to the vertebra at the back side dorsally, but ventrally they are free, hence they are said to be the floating ribs. Remember guys, 8, 9, 10 are false, 11 and 12 are also false. That does not mean the floating ribs are true ribs. Total number of false ribs are 5 pairs. In that vertebrochondral 3 pairs, floating ribs are 2 pairs. This sternum, 12 pairs of ribs, 12 thoracic vertebrae put together forming a rib cage. Rib cage. So, with this I have completed the axial skeleton. The next topic is appendicular skeleton. Appendicular skeleton consists of girdles and limbs. Let us talk about pectoral girdle. This girdle is called pectoral, which is supporting the upper limb. Each half of pectoral girdle I will explain. Each half of pectoral girdle consists of 2 bones. One is clavicle, second one is scapula. See, these are the ribs, amma. Back side of the ribs there is scapula. See, scapula is a triangular flat bone located on the dorsal side of the rib cage, not on the front, on the back side. It is extending between second rib to the 7th rib, second to 7th, and it is flat in nature. This is the front side of the scapula. If you see the back side of the scapula, I am showing you the back side of the scapula. On the back side of the scapula, there is a flat extension, there is a spine. This spine extends as a part known as acromion process. So, this is a spine guys. Spine is located on the dorsal side. It extends as a flat a component called acromion process. See this? This is acromion process. Below the acromion there is a cavity. This cavity is called glenoid cavity. This is the first bone of the upper arm. This upper arm is called humerus. The head of the humerus is fixed into this cavity, forming a ball and socket joint, which is nothing but shoulder joint. Come again. Scapula is located on the dorsal side, flat triangular bone extending between 2nd to 7th rib. The spine present on the dorsal side of the scapula extends and forms a flat component called acromion process. Below the acromion there is a glenoid cavity. Glenoid cavity along with the head of the humerus forming the ball and socket joint, nothing but shoulder joint. This is about scapula. Let us talk about clavicle. Clavicle is called collarbone. It has 2 curves, curve 1, curve 2. I already told you guys, laterally the clavicle is attached to the acromion process of the scapula. Medially the clavicle is attached to the sternum. medially attached to sternum. See, sternum belongs to axial skeleton. Acromion process of scapula belongs to appendicular skeleton. So, clavicle is said to be connecting a part of axial skeleton with the part of the appendicular skeleton. And this is a thin slender bone which is subjected to easy fractures during accidents, horizontal slender bone. So, this is about appendicular skeleton, and appendicular skeleton is supporting upper limb bones. So, we can divide the upper limb into three parts like upper arm, forearm and hand. The bone of the upper arm is called humerus. On the lateral side of the humerus, there is a slight extension present known as a deltoid ridge. This is there in practical manual. And humerus, one end is forming the ball and socket joint, other end of the humerus with the radius and ulna forming a joint called hinge joint. And the forearm consists of two bones, one is radius and second one is ulna. How can you recognize in a picture where is radius and where is ulna exactly? You remember always guys, towards the thumb side, whatever the direction of hand, whether it is pronation or supination, whatever it is, towards the thumb side there is a bone called radius. See, for this hand this is radius. If I turn like this, again for thumb side is radius only. So, radius and ulna is very well developed compared to the radius. Next is hand. Hand consists of three portions, wrist, palm and fingers. The wrist consists of carpals, there are 8 carpals present. The palm consists of metacarpals, there are 5 metacarpals. And the phalanges are 14 in nature. Thumb consists of 2 phalanges, remaining 4 fingers consists of 3 phalanges in each. 14 phalanges plus 5 metacarpals, 19 plus 8 carpals, 27, 1 ulna, 1 radius and 1 humerus. 30 bones are present in each upper limb. Next is a pelvic girdle. See the next one. This is pelvic girdle. The pelvic girdle is made up of 2 bones called coxal bones. This is one coxal bone guys, one side coxal bone, other side one more. So, this is the coxal bone. Each coxal bone is formed by the fusion of 3 bones, ilium, ischium and pubis. And we shouldn't consider ilium, ischium, pubis are separate bones. To all the 3 put together fused to form a single bone called coxal. If they ask any question like how many bones are present in pelvic girdle, the answer should be 2 only. Pelvic girdle is made up of 2 bones, they are called coxal bones, having a broad shield like structure called ilium, then ischium and pubis. See this, a part of pubis, part of ischium, part of ilium forming a cavity called acetabulum. This acetabulum with the head of a thigh bone called femur forming a ball and socket joint known as hip joint. See, there is a shoulder joint, there is a hip joint, both are coming under ball and socket. Both are allowing the free movements, they are said to be the synovial joints. The acetabulum is formed by all the 3 components. And when we see the ventral location of this pubic bones, this is one pubic bone and I would like to draw another pubic bone here. Between 2 pubic bones, there is a cartilage present known as pubic symphysis. So, the 2 pubic bones ventrally joined by a cartilage called pubic symphysis. Pubic symphysis is also the example for cartilage in a joint. It is made up of fibrous cartilage. So, this is about pelvic girdle. Next, what is the bone of vertebral column that is fixed into this pelvic girdle? Nothing but sacrum. Sacrum is fixed, and finally coccyx. Next, pelvic girdle is supporting the lower limb bones like thigh, shank and foot. Thigh is having a bone called femur. Femur is the largest bone in the body. It is forming the ball and socket joint at one end, proximal end. It is forming a hinge joint at the lower end, nothing but knee joint. After femur, there is a triangular bone on the ventral side called patella. Patella is said to be the kneecap, which is a sesamoid bone made up of tendons. Next, the shank consists of 2 bones, tibia and fibula. See this, tibia is towards the medial side, fibula is towards the lateral side. And tibia is stronger compared to the fibula. The weight bearing capacity is done by the tibia. When it comes to the foot, foot consists of ankle, foot consists of instep and phalanges. The ankle is made up of torsals. There are 7 torsals. The torsal which is present at the ankle region towards the back is called a heel bone. Heel bone is known as calcaneum. Calcaneum is the largest ankle bone. Next, the instep consists of 5 metatarsals followed by phalanges. Phalanges are 14 in number. Big toe is having 2 phalanges. Remaining are having remaining 4 toes are having 3 phalanges in each. So 14 phalanges plus 5 metatarsals. 14 plus 5, 19. 7 tarsals. 19 plus 7, 26. 1 fibula 27, 1 tibia 28, 1 patella 29 and 1 femur 30. So each lower limb is also having 30. And here I want to tell you a point. There is a corresponding bones between upper limb and lower limb. For example, if I take humerus, what is the corresponding bone in the leg? Femur. Radius ulna corresponding bones are tibia and fibula. Carpals corresponding bone tarsals. Metacarpals corresponding bone metatarsals. Phalanges. But there is no corresponding bone for patella in the upper limb. There is no corresponding bone for patella in the upper limb. So with this the pectoral the appendicular skeleton is completed. The next topic is joints. Joints are essential for all types of movements involving the bony parts of the body. It's not like joint is made up of only bony. Joints are made up of bone and cartilage. There are 3 major types of joints, guys. Fibrous joints, cartilaginous joints and synovial joints. I already told you the sutures between the cranial bones comes under fibrous joints. That joint is made up of dense fibrous tissue. It is the best example for immovable joints. The second one, cartilaginous joints. This also I gave you. Between the centrum to the adjacent vertebrae, there is a cartilage called intervertebral disc. Intervertebral disc is the best example for cartilaginous joint. Cartilaginous joints are allowing the limited movements of the body. Next, when it comes to the third category, that is most important. Even pubic symphysis comes under cartilaginous joint. The third category is synovial joints. Regarding the synovial joints, they are freely movable joints. Synovial joints are freely movable. See this? The basic structure of synovial joint I just want to mention here. Assume it is a bone. This one is another bone. These 2 bones are held together by a capsule. This is the capsule. The capsule is made up of 2 layers, outer fibrous capsule and inner membrane is called synovial membrane. So holding the 2 bones together, between the 2 bones there is a cavity filled with a fluid called synovial fluid. Because of synovial fluid, there will be free movement at these joints. Outer fibrous capsule, inner synovial membrane secretes a synovial fluid. Next, a synovial membrane is made up of areolar connective tissue. At the tip of each long bone there is a cartilage. It is hyaline cartilage. It is said to be the articular cartilage and it allows free movements. All the synovial joints are freely movable. The examples for the synovial joints includes, listen carefully, first is ball and socket type. The examples for the ball and socket joints are shoulder joint and hip joint. Second one is hinge joint. The elbow joint and knee joint are the examples for the hinge. Even interphalangeal joints, which is there in the exercise part of NCERT textbook, they also comes under hinge. Hinge allows flexion and extension movements. Between the atlas and axis there is a joint called pivotal, allows rotatory movements. Next, between carpal and metacarpal of thumb we have a joint called saddle joint. And between the carpals, between the tarsals there is a gliding joint. That means the 2 flat carpals can slide upon each other in different directions, forming a gliding joint. So examples of synovial joints: ball and socket, shoulder and hip, hinge joint, elbow joint, knee joint and interphalangeal joints, pivotal, atlas and axis, atlanto-axial joint, saddle, carpal and metacarpal of the thumb, gliding intercarpals, intertarsals. And the last topic is the disorders of the musculoskeletal system. The first one is myasthenia gravis. Myasthenia gravis is autoimmune disorder. I already gave you the picture of excitation type. See, between the motor neuron and muscle there is a neuromuscular junction. Neurotransmitter is released, it will bind to the receptors. In myasthenia gravis, neurotransmitter is released, no issue with that. But the receptors present on the sarcolemma will be blocked by the our own antibodies. When they are blocked, though neurotransmitter is released, it will not bind. When it is not binding, there is no impulse transmission to the muscle will not contract. that leads to muscle weakness, muscle fatigue and paralysis. It is a neuromuscular junction problem. The next one is muscular dystrophy, progressive degeneration of skeletal muscles, mostly due to genetic disorder. One example for that genetic disorder is muscular dystrophy. Duchenne muscular dystrophy is a X-linked recessive disorder, where there is a problem with a protein called dystrophin. Tetany, wild contractions in muscle due to low calcium in body fluids is called tetany. And we can also give the definition for the tetany is lack of relaxation between successive stimuli in sustained muscle contraction is known as tetany. Arthritis means inflammation of joints causing pain, age related, the treatment like hip and knee replacement. Osteoporosis is also age related disorder characterized by decrease in bone mass due to resorption of calcium from bone to the blood. It is caused by the osteoclast cells. It is more common in females at post menopause due to the decreased levels of estrogen. Whenever estrogen is decreased, that will not have any inhibitory effect on the osteoclast. Osteoclast cells causes the bone resorption, causing the decrease in the bone mass, and which will cause easy fractures. Of course, calcium deficiency also responsible for the osteoporosis. Vitamin D deficiency also responsible. Finally, the gout. Gout is also inflammation of joints, but not due to erosion of cartilages, age related like arthritis. Here the inflammation of joints is because of accumulation of uric acid crystals. When more uric acid is produced by purine catabolism, if kidney is unable to send it out, it will straight away go and deposit at the joints in the form of sodium monosalts. They are nothing but uric acid crystals. It causes severe pain. This condition is said to be gout. So with this, I have completed the locomotion and movement chapter in detailed description of muscle and all the components, skeleton and the related components. Thank you.